Again, a review of interphase. Interphase is the cell just being a cell. It's not dividing, and it consists of G1, S, where we replicate all the chromosomes, and G2 phases. G1, we have the major portion of growth. S, we replicate DNA, and in G2, the chromosomes are coiling, the cells preparing for division. The centrioles replicate, and tubulin synthesis begins, so we can maneuver the chromosomes around in the cell. Let's take a closer look at the centromeres. The centromeres are the point of constriction on each chromosome. Sometimes they're located in the middle section, but sometimes they might be located more towards the end of an arm. Each sister chromatid has a centromere, and sister chromatids stay attached to the centromere by this cohesin molecule. On the outside of the centromere, we'll also see a region called the kinetochore. The kinetochore is the attachment site for the microtubules that are going to orchestrate the movement of chromosomes during mitosis. So M phase, or mitosis, is divided into 5 different phases: prophase, prometaphase, metaphase, anaphase, and telophase. We'll begin by looking at prophase. What's most important here is that we keep track of how many chromosomes, chromatids, so on and so forth there are, using the language that we learned in the previous lecture section. In prophase, the individual condensed chromosomes first become visible under a light microscope. The condensation continues throughout prophase. The spindle apparatus coming off the centrioles will begin to assemble, and the centrioles begin to move to the opposite ends of the cell, or opposite poles. Now, plants don't have centrioles, so they wouldn't be seen. The asters are this radial array of microtubules that we see around the centrioles. Again, we don't see those in plants because they don't have centrioles. The other thing that happens during prophase is the nuclear envelope breaks down. As you try to understand what's going on in each of these phases, it makes most sense to step back and understand what needs to happen. If you understand what needs to happen, then you can fit the appropriate events into the appropriate phases. So in the whole scheme of things, the nuclear envelope needs to break down and the chromosomes need to line up along the middle so that we can separate them and then wrap them back up in nuclei. So in prophase, we've started to break down the nucleus because it makes sense, and we've started to assemble the puppeteers. That's what I think of the centrioles as puppeteers that will attach themselves to the chromosomes and move them about inside the cell. We begin prometaphase after the nuclear envelope has disassembled. The microtubules will now attach to the chromosomes. There are 2 types of microtubules. There are ones that reach out from the centrioles and connect to the kinetochores. And then there are other ones that are connected to the opposite poles of the cell. These microtubules work in conjunction to elongate the cell, as well as to eventually pull the sister chromatids apart. So at this point, each sister chromatid is connected to opposite poles of the cell, so that when these microtubules shorten, they can pull apart. In prometaphase, the chromosomes begin to move around towards the center of the cell as the puppeteers, the centrioles, start pulling on the strings. In this process, you'll see an assembly and disassembly of microtubules as they lengthen and shorten and manipulate the chromosomes into their proper locations. Meta means middle. So it's the middle phase of mitosis, and it's where all of the chromosomes line up along the middle of the cell in an area that we call the metaphase plate. However, there's not an actual structure there. It's a theoretical place. The metaphase plate you can see here is where all the chromosomes are lined up. It's the future axis of cell division. It's where the cell will pinch and divide. So let me ask you a quick question here. How many of what are lined up down the center of this cell? There are a number of different ways of saying it. So how many of what are lined up here along the metaphase plate? We have 4, definitely, something, right? We have 4 But what are they? Are they chromosomes, chromatids? I'm going to say 4 chromosomes. What other way could we say this? More correctly would be 4 replicated chromosomes. Or how about this? 4 pairs of sister chromatids. Lots of different ways to say the same thing, but just so we can continue using the language we've previously learned. In anaphase, the centromeres split. That is, the sister chromatids are pulled away from each other. And when sister chromatids separate, they can no longer be called chromatids. So now they're called daughter chromosomes. All of the cohesin proteins are removed from between each pair of sister chromatids. This is what allows them to split. And the microtubules pull the new daughter chromosomes towards opposite poles of the cell. There are 2 forms of movement going on with these microtubules. There are kinetochore microtubules that are attached to the centromere of each of the chromosomes. And they're shortening and pulling the chromosomes towards the pole. However, you'll notice there are also another type of microtubule that do not attach to the chromosomes themselves. They simply push on each other, and as they add length to their fibers on this inside end, the cell elongates and the poles move away from each other. Those are called polar microtubules. So we want to understand the difference then between the kinetochore microtubules and the polar microtubules. Polar microtubules push on each other in order to lengthen the cell, and kinetochore microtubules attach to the kinetochores of the chromosomes and pull them apart. In telophase, this is where we see the spindle apparatus disassembling, because after all, it's done its job now. And the nuclear envelope has to form again because we need to package these chromosomes back into nuclear envelopes. So a new nuclear envelope will form around what we were calling each set of sister chromatids, one of the copies. However, we now call them chromosomes because they're not attached to each other. The chromosomes will begin to uncoil and will move back towards being ready for interphase. As the nucleus forms and the chromosomes begin to uncoil, we'll see a nucleolus, which is the darker staining region in the nucleus. So at this point, we've completed nuclear division. The nuclei have split. However, the cell, you'll notice, is still one unit. So mitosis, this is a key thing to remember, mitosis does not include cytokinesis. So here's a question. How many of what are in the cell here at telophase? Just to test that we know our language. So right now in this cell, I'm going to say there are 8 chromosomes. However, if the question says in each new nuclei, because in this phase we're beginning to wrap these up in a new nuclei, you would have 4 chromosomes in each new nuclei, but 8 in the cell in total. If we want to be very specific, we can say 8 non-replicated chromosomes. So cytokinesis is actually when the cell divides. So mitosis is nuclear division and cytokinesis is cell division. It's when the cell cleaves into equal halves. In animal cells, we'll see a constriction that's formed by actin filaments, as you can see in this figure over here. And it produces a cleavage furrow. And literally, as if you were to tie a string around a water balloon, it would pinch all the way through the middle. However, plant cells have to also take care of cell walls. The cell plate that will eventually be a new cell wall will form between the nuclei. Little vesicles will deliver materials all the way down the center of the cell in order to build the new cell wall. Some fungi and protists don't have the nuclear membrane dissolve at all. Mitosis occurs within the nucleus, and then division of the nucleus occurs with cytokinesis. So the nuclear envelope doesn't break apart as we see in prophase of animal mitosis. So let's review quickly. We'll begin with a cell that's in G1, let's say, and we need to go through S phase in order to get to G2. Now, let's say 2n equals 6 for simplicity. What do the chromosomes look like during G1? In G1, we have 3 pairs of chromosomes. We have 2 red ones, we have 2 yellow ones, and we have 2 blue ones. So there are 3 distinct different chromosomes. So the n is 3. 2 times n equals a total of 6 chromosomes. After we go through S phase, what do these chromosomes look like? During G2, we have still 3 pairs of chromosomes. However, they're different. Let's say in G1, the chromosomes weighed, all the DNA weighed 10 picograms. How much would the DNA weigh in G2? Right, we would have 20 picograms. Now let's consider meiosis. Now let's consider mitosis. We move on into M phase. I'm just going to say we're in metaphase of M phase, so we can keep this straight. What does the cell look like? What does the nucleus of the cell look like with respect to the chromosomes? During metaphase, homologous chromosomes will line up single file down the center of the cell. So there are how many of what on the metaphase plate? There are 6 replicated chromosomes. How much does it weigh? Still 20 picograms. And then what happens in anaphase? Each of these chromosomes pull apart, and they're moving to opposite poles of the cell. What do we have inside the cell now? We have 12 chromosomes. And then when we move into telophase, we have 2 new nuclei, each with 6 chromosomes, unreplicated. Hopefully this helps you out with your diagramming this week.